I think there are very few risks. Everyone who donates does get a consent form, and the biggest risk that is in all of those consent forms is the potential risk that private medical information could somehow get out into the wrong hands. Uh, that's a theoretical risk. I can think of very, very few cases in the country, frankly, where that's happened. There are a lot of safeguards, and the way at least we code all of that information is not using patient names or initials. We don't even use social security numbers or birth dates. Everyone gets a completely random number, and all the analysis is really done based on those numbers. It's an interesting point because it's not uncommon that we will get asked by patients, well, what did my tumor show? What did you find? Do I have this? And the truth is that we really can't always go back and tell people that for exactly that reason.
If you have a colonoscopy and they take a lot of little tumors out of your colon, can they send those tumors to your?
Did everybody hear the question?
Yeah.
The question was, if you have a colonoscopy and you have many tumors removed, is there a way to contribute those tumors to the bioconsortium?
I think for some of the reasons we were just talking, privacy and making sure that everyone has proper consent, it turns out it can sometimes be difficult for people simply to send their tumors and have them analyzed. There are many ethical review panels that have to look at this overall procedure really to protect patients and make sure that privacy is not invaded in any way. So I would say that the best way and surest way to contribute to one of these databases would be to at least get evaluated at one of the centers, sign the consent form, and just have everything be above board in that way. I think it's great that there will be more centers now with the ability to do this for neuroendocrine tumors.
My question too was, would a regular lab be able to really diagnose a carcinoid tumor?
You know, 10 years ago, I would have said I might have some concerns about that. There is so much more interest now in carcinoid and so much more awareness that I frankly think that's much less of a concern than it was. In most cases now, at least the pathologic diagnosis is being made very appropriately. There's still delays in diagnosis, I would say, because this can be a very hard condition to diagnose early on.
Thank you, Dr. Kulke. Now we have Dr. Matthew Meyerson, who is the director of the Center for Cancer Genome Discovery at the Dana-Farber Cancer Institute and the Broad Institute. Dr. Meyerson is also currently being funded by the Caring for Carcinoid Foundation for a large-scale genome study of carcinoid tumors.
So, yeah, I want to talk about the idea of going from genes to drugs, and thereby being able to really develop new treatments for carcinoid that are based on the genetic mutations found in the disease, as Dr. Kulke was referring to. Oh, I forgot to turn on my mic. Okay, I have it on my laptop, but okay. And so what I'd like to do today is just talk a little bit first about, what is a gene, what is a cell, and how are genes altered in cancer? How do these altered genes cause cancers and cause tumors such as carcinoid? And then how is this relevant to cure? And just want to mention at the beginning, formally, a couple of conflicts of interest that I have research and or consulting support from Novartis, who manufactures Gleevec, and from Genentech, which manufactures Tarceva, two drugs that I'll mention briefly. And I'm also an inventor on a patent for using the EGFR gene for cancer diagnosis. Okay, so why do we care about cancer-causing genes? And I think the reason is because cancer is caused by changes in genes and in the genome, and I'll talk more about that later. The second reason is to understand what causes cancer so that we can think in the right way about this. And this could affect prevention and surgery and even potentially nutritional ways to prevent cancer. And finally, and probably most important, to treat cancers and other tumors by actually blocking the activity of cancer-causing genes. And this has worked for many diseases. So some examples of that recently are imatinib or Gleevec for chronic myeloid leukemia that has activation of the ABL gene. Trastuzumab or Herceptin for breast cancers that have activation of the ERBB2 gene. And erlotinib or Tarceva and gefitinib or Iressa for lung cancer that has activation of the EGFR gene. And so this is a kind of example here of the promise of gene-targeted cancer therapies. And if we think about the need, overall, cancer kills more than 500,000 people in the United States each year. And cytotoxic chemotherapy is highly toxic and often not very effective. In contrast, the hope is that selective therapies against altered cancer-causing genes can be highly effective and can exert fewer side effects. And so this is an example. It's a patient with lung cancer who has an EGFR mutation in her tumor. And you can see before treatment, there are multiple nodules. Again, I just apologize, it's not full scale here, so it's harder to see. But you can see multiple nodules here in the right side of the lung, the right side because this is flipped. And within 2 months of treatment, these nodules have completely disappeared. And with, in fact, it's been recently shown very convincingly that patients whose tumors have EGFR mutations benefit specifically from Tarceva treatment or Iressa treatment, whereas those patients whose tumors do not have the mutations do not benefit in the same way. So I think this is very good evidence that we need to both identify the genes that are underlying the development of cancer and that we need to actually diagnose these eventually in patients. And there are diagnostics available for a few specific cancers and a very few specific gene mutations. But I think over the next couple of years, we're going to see this become much more general. And the ability, in the sense that the woman was asking after Dr. Kulke's talk, can we actually use tumor tissue for diagnostics on the DNA level? The answer is not yet, but hopefully very soon. This will become widespread. So I just want to talk about what is a gene, what is the genome, and what is a cell. So genes are the instructions to make proteins. And proteins perform the basic activities of cells. And billions of cells make up our bodies. And I'll show you a little bit more about what a cell is. And genes are stretches of DNA, which is our genetic code. And they're used to make RNA and then protein. And genes are units of inheritance that are passed from parent to child and also from cell to cell. So if we think about the genome, DNA is the genetic code. It has 4 letters, A, C, G, and T. So I'll talk about where we were, which is so DNA is made up of 4 letters. There's A, C, G, and T. And so you could think, well, how do you actually, let's go back 1 slide. And I guess this is pretty good. There we go. 4 letters, A, C, G, and T. And some stretches of DNA code for proteins, some of them for chromosome structure, and some DNA structures control when or where proteins are made. And our genome is made of 6 billion DNA letters. 3 billion for each of us come from our mother, and 3 billion come from our father. And they're organized in 46 chromosome strings, and they include around 20,000 genes. So if we can advance, every cell contains all 6 billion letters of DNA. And cells are the units of function in the body. They're very highly specialized. So there are blood cells, muscle cells, brain cells, germ cells like egg and sperm, skin cells, and then there are the endocrine cells, which are the precursors of carcinoid. Each cell, as I mentioned, contains a complete genome. There are around 100 trillion cells in the human body. Some cells can divide and make daughter cells, normally in a very highly controlled manner, which is programmed in our DNA from birth. and other cells can't. And I just want to say this is a picture of cells here, and here the DNA is in the middle, it's stained green, and the membrane covering around the outside is stained red. So how is a cancer cell different? So cancer cells are partly like other cells. They still have a lot of the features like shapes and proteins of related cells. So stomach cancer cells look related to the stomach lining. Carcinoid cells look related to other neuroendocrine cells that secrete hormones. But they have different growth patterns than normal cells. They divide when they shouldn't, and they grow in places where they shouldn't. That's what's called metastasis, when a tumor grows out of its original place and moves to a new place in the body. And if we could look at the next slide, the cancer genome is different too. Sometimes I actually quote Leo Tolstoy, who started Anna Karenina, and he said, all happy families are alike, but every unhappy family is unhappy in its own way. And in the same way, basically the genome of all normal cells is pretty much the same. In fact, in our body, almost every normal cell has almost exactly the same genome. And even between people, almost all cells have, between people we have almost the same genomes. But cancer cells can have very different genomes, and every cancer is a little bit unique, but they also have shared features. So the DNA of cancer cells is different from the normal cells in the same person, or mutated. And a mutation means a change in the DNA sequence or structure, and it implies some kind of functional significance of that change. And the genomes of cancer cells actually can change as the cancer evolves, as the disease gets more severe or patients become resistant to drugs. So the disease in the first tumor is not necessarily the same genome as the tumors that's involved. So cancer patients may be born with DNA sequence variations. In fact, we're all born with variations. These variations affect the whole body. They're called germline, and it may be either inherited from the parents or maybe new mutations in the patient. And these variations can actually increase the risk of cancer. And we probably all have some variation that increases or decreases our risk of particular cancers. On the next slide, the other thing that happens, though, in cancer is that there are changes that happen somatically. They're only in the cancer, and they're not in the rest of the body. Only in the tumor, not in the rest of the body. And so these include mutations, which are, I mentioned these 4 letters, A, G, C, and T, so you get changes. Each one of these changes shown here could be cancer change causing. Amplifications or deletions, where you get extra copies in an amplification of a gene, or too few copies. Translocation, chromosomes break and rearrange. We normally have 46 chromosomes in every cell, 23 from our mother, 23 from our father. In cancers, these numbers could be different. And finally, infections. So each of these are different events that can cause cancer. They're all occurring on the DNA level. On the next slide, because the cancer genome is different from the normal genome, cancer cells have different survival requirements from normal cells. So in principle, we can find we can find drugs that will kill cancer cells, but have much less effect on normal cells. And just to make an analogy, this is very similar to the approach to kill infectious diseases. So for example, penicillin is a drug that blocks the synthesis of the cell walls of bacteria. Our cells don't have cell walls. So we can use penicillin very effectively. It will kill bacteria that are in our body, but it won't kill our own cells. In principle, once we understand the genetic differences between normal cells and cancer cells, we can do the same kind of thing for cancers. So there are 2 types of cancer genes. There are oncogenes, which promote cancer growth. They become hyperactive in cancer. And there are effective drugs that target oncogenes, like imatinib or Gleevec that I was talking about before. There also are tumor suppressor genes. Can we back up for 1 second? They suppress cancer growth, it should say. They lose activity in cancer, and there are no effective drugs yet for this class. If we can go forward now. So this is a little cartoon to illustrate them, and this is an idea that comes from my postdoctoral advisor, Bob Weinberg. He said that oncogenes are a little bit like the accelerator of a car. Normally, they're giving it a little push to grow, but in cancer, they become hyperactive, and you get lots of extra activity. So tumor suppressors, in contrast, are like the brakes. Normally, they stop cells from growing abnormally, but again, in the cancer, they're broken, and you've got no block to growth. So looking at this slide, when I was a kid, I actually dreamed about being an architect. But if you look at my artistic talent, I think this is part of why I decided not to become an architect and to go into medical research. So the next slide, I just want to say, talk about the Carcinoid Genome Project, which is our effort to learn about all of the genes that are altered in carcinoid. And so the goals originally are to read the DNA sequence of 2,000 genes in 48 carcinoid tumors and matched blood from the same patients. Then to evaluate the presence of cancer-specific mutations in this DNA. And then finally, we want to perform experimental studies of these cancer-specific mutations to determine whether they are targets for drug therapy. So on the next slide, we say, why do we think we can do this project now? First of all, there's a technology revolution in genome analysis that's going on right now. Second, as Dr. Kulke was mentioning earlier, there's a really great carcinoid tumor bank that's being sponsored by the Caring for Carcinoid Foundation. And finally, again, as Dr. Kulke was describing, we've seen clinical trials of some new targeted therapies, and they're showing great promise. And so for this reason, I think there's a lot of hope for finding new drugs. So the next slide, I'll describe a little bit about the technology revolution. And some of you may be familiar with Moore's Law. Gordon Moore was one of the founders of Intel, the big semiconductor company. And he said that integrated circuits get twice as powerful every 2 years. And it's actually turned out to be true. And because of this, we've seen this incredible revolution in computers and communications over the last 50 years. And with genome analysis, this is actually happening even faster. It was, for many years, better than twofold improvement a year. And last year, this year, and probably next year, it's more like a tenfold improvement every year. So we can just, the genome technology is advancing by leaps and bounds. And what are called next generation sequencing technologies allow us to read millions of DNA sequences at once and discover mutations, copy number changes, translocations, and infections, all those different examples of genome alterations that I was describing. And this is, let's just show one more thing here. This is just an example of one of these technologies. It's a sequencing technology from a company called Illumina. And what you can see here, red, yellow, green, and blue represent each of those 4 letters, A, C, G, and T in DNA. This is more of a faraway view. And this is like a more faraway view or a visible view. And the penny here is shown for scale to show what the size is. This is not yet the cost of sequencing one human genome, but at the rate things are going, it might be pretty soon. So finally, I just want to talk about, on the next slide, about the carcinoid sample collection. Thanks to CF, and other support, physicians and scientists have been building collections of frozen carcinoid tumors. And for the project that we're doing, there's an international network. Drs. Kulke and Shiv Dasani, both of whom you've seen speak today, Drs. Yao and Rashid from MD Anderson Cancer Center in Houston, and Dr. Sylvia Asa from the Princess Margaret Hospital in Toronto. And in terms of hope for finding new drugs, our DNA sequencing efforts are going to be focused on genes that are known targets. We will then test the mutated genes to determine whether or not they do promote cancer. We will look for mutations that might predict responses to known drugs, which could rapidly be moved into treating patients, as Dr. Kulke described with the example of Sutent. And finally, we will make our data publicly available